So for the cell-mediated response, what we're looking at here is what happens when a pathogen is hiding or reproducing inside of cells. So here let's take a virus, and a virus is hiding within a cell and it's replicating. Remember, a virus needs a host. So when there's a virus hiding inside of a cell, it would be ideal if we can stop the replication of that virus so it doesn't spread throughout the body. So all cells, or most of their body cells, have what's called a major histocompatibility complex. We call this MHC for short. Now on most of your body cells, it's a class 1 MHC molecule. And so this class 1 MHC molecule is going to take a piece of the virus that is, or the pathogen that is replicating within the cell, and it's actually going to present a piece of that antigen or virus on its surface. Now this is like saying, hey, look what's replicating within me. And so class 1 MHC molecules, they display foreign antigens that were made within the cell on its surface. So that's the key. This is saying, hey, this is being made within me. It's not engulfing and taking it in by phagocytosis. Instead, it's saying this virus has replicated within me and I'm showing you what's inside my cell. So this attracts cytotoxic T cells, or killer T cells. Now, the way killer T cells work is that they are activated by cytokines, which come from helper T cells, which we're going to talk about in a few minutes. But here, once the cytotoxic T cell has become activated by cytokines, and so really when we say activated, what that means is that the killer T cell has like a receptor on its surface, like a protein receptor, and the cytokine attaches and starts a signal transduction pathway where the cell response of the killer T cell is to produce proteins. So these proteins are called perforin because they perforate or put holes inside the virus-infected cell. So these proteins are going to form rings within the cell membrane like this. Now the rings that are put inside the cell membrane of the virus-infected cell result in cell lysis and cell death. And so now that virus-infected cell is dead, and the virus can no longer replicate and spread. So any debris or virus particles or pathogen particles that are present will be cleaned up by white blood cells. So there's a better picture of cell lysis. So cytotoxic T cells kill pathogen-infected cells by putting holes in their cell membranes, resulting in cell lysis. Okay, so that's the cell-mediated response. But how did the cytokines get there? Where did they come from? Now here you have a dendritic cell, or a macrophage, that's engulfing a pathogen. And when it engulfs the pathogen and it degrades it within the vesicle inside, using lysosomes and lysozyme, a piece of this antigen is going to get taken by a class 2 MHC molecule. And it's going to present that antigen on the surface of its cell. So when this macrophage or dendritic cell presents the antigen on its surface, we call this cell an antigen-presenting cell. And so now this is going to attract helper T cells. And that's exactly what they do. They help. So this helper T cell is now activated because it attached to an antigen-presenting cell that is displaying a class 2 MHC molecule. Now, when we look at class 2 MHC molecules, they're showing antigens from foreign materials that were internalized or taken in by endocytosis or phagocytosis. This is different than the class 1 that was being replicated within. Okay, so then the macrophage, or the antigen-presenting cell, activates the helper T cell, and now that helper T cell can help both the cell-mediated and the humoral. So when we talk about, like, well, how does it help the cell-mediated, that's back on this slide. These cytokines right here came from a helper T cell. So when we look at the humoral response, though, in this situation, remember when the B cell, out of the million B cells you have, one of them has antigen receptors that match the antigen on a pathogen. So when we talk about clonal selection, that's kind of like the selection part. Anyway, this B cell needs some kind of information to signal it to divide. So with that, that's where the helper T cell comes into play. Once the helper T cell has been activated, it's going to then go over to the helper T cell where it will release those cytokines, and it's going to activate the B cell using a signal transduction pathway, and the cell response is for the B cell to go through mitosis. So the B cell starts to divide, make its clones, its plasma cells, and its memory B cells. And so that is how the helper T cell helps both the humoral response and the cell-mediated response. Okay.